Okay, so today I'm going to talk to you about a new statistical framework that we developed in the lab to do single-cell differential expression meta-analysis in population studies. And the main question that we are trying to tackle in the lab is how does human diversity emerge? And if we think about some of the main demographic traits or differences and the way we try to tackle this question is by looking at the transcriptome and understanding how the transcriptome is regulated. And then we go from transcriptome regulation to human phenotype, human diversity, and sometimes disease. Okay, and for this particular study, we asked a very simple question. And this question is, what are the main drivers of human transcriptome variation? So we think about the transcriptome and we want to understand human diversity. It would be ideal to understand what is the relationship between these demographic trait phenotypes to gene expression variation. And I would like to take 2 seconds to think about this. So if you take your gene expression data set, your favorite one, and you think which one of these individual traits or demographic traits is going to have the largest impact on gene expression, we certainly did not know the answer to this question when we started the project, but we thought that the best way to answer it is to use the genotype tissue expression project, because in this case we had information about 1,000 individuals, 46 different tissues, 10,000 samples. And for all of them, we knew their genetic ancestry, their biological sex, age, and so on. And we really didn't know which one of these individual traits would have the largest impact on gene expression. All right. So this is the distribution. We have genetic ancestry related to European and African American individuals, male, female, and age spanning from 70 to 80 year old, and also BMI information for all the donors. All right, so what is the individual trait that has the largest impact on gene expression variation? And the way we did to answer this question is to do a hierarchical partition approach, where basically the goal is to quantify the amount of expression variance that can be explained by each demographic trait, and it identifies the most likely causal factors while elevating multicollinearity. So basically, we took the residuals, we regressed out the batch effects, and we tested the contribution of each of these demographic traits to gene expression variation. And these are the results. So in biology, the answer is never, easy. And in fact, what happens is that this is completely tissue dependent. So the contribution of each demographic trait is very different across tissues. For example, in breast, for example, the contribution of sex is very large between males and females. I mean, we know this, right? Everyone knows that. But what's interesting about this is that we can know what are the genes exactly that are driving these effects. And then in pituitary, for example, which is involved in metabolism, but also in regulation of lactation, we also see the same trends. We also have some tissues like adipose, for example, where we see that BMI has a strong contribution, and muscle, where all the 4 demorphic traits have a similar contribution. And this is, like I said, most of these findings make sense when you think about it, but the interesting part is that you can know which are the genes that are driving these effects, okay? So in this plot that looks very complex, but basically in the x-axis we have the tissues, and in the y-axis we have the variants explained by each gene and demographic trait. And we can now find, for example, genes that their variance is explained by one demographic trait. So here, for example, the genes ACKR1 is the malaria receptor. So these genes has a 40% of its variant explained by genetic ancestry, and it's related with the fact that African American individuals are less susceptible to get malaria infection than European American individuals. So we are going from this transcriptome analysis to understanding phenotypic differences between individuals. All right. So I'm not going to go more in depth with this study because this is published data. We also looked at alternative splicing and how these demographic traits explain differences in alternative splicing variation. The answer that we got was completely different to the one that we got for expression. And also we looked at clinical traits like diabetes, which we also found that the gene expression variation the answer is quite different. So I encourage you to take a look at the publication. But what I want to talk to you about today is that a lot of the findings that we had for this study were really confounded, if you will, by cell type composition changes, right? So a lot of the differences that we saw could be related to gene expression and cell type composition changes. So we decided to start a project where we're going to look at cell type single cell resolution. And when we were thinking about how can we understand sex and age differences and the contribution of sex and age in gene expression variation at single cell resolution, we realized that there were many data sets out there available, specifically for PBMCs. And the sample size were small to moderate. And this is and they were coming from groups all over the world. And this is a similar problem to the people in the equatorial communities facing. So to do equatorial analysis, similarly to do differential gene expression and understanding individual variation, you need large cohorts, okay? And in PBMC, there's starting to be some cohorts that were like, we're talking about 80 to 100 donors each, and they were generated in different groups. So we decided to join the consortium because to join forces, their interest or the interest of the consortium is to understand how genetic variation affects gene expression. Our interest is to understand how gene expression varies with sex and age. And the approach that the consortium decided to use, and we are also using, is to do a meta-analysis. Why do we want to do a meta-analysis? Well, if you think about having 28 different groups generating data and sharing data, can be a nightmare because there is a lot of privacy issues, and it becomes really, really hard. So if you do a meta-analysis where the goal of the meta-analysis is that you develop the pipelines, you distribute the code through the different groups, the groups run the algorithm, and they bring back the results, and then you meta-analyze these results. It gets rid of the problem of data sharing. It also, you don't need to do, imagine, some groups are going to be very fast, some groups are going to be slow, some group is going to join the consortium. So every time that someone new generates new data, if you're doing reanalysis, you need to rerun everything. Whereas if you do a meta-analysis, right, you just need to incorporate the summary statistic and it's done. And in the context of working with, like I said, many different groups, this makes your life easy. And then, of course, doing meta-analysis has lower running times and memory consumption. And we're talking here about millions of cells and hundreds of donors, if not thousands, if you put all the data sets together. So many of the methods that are out there to do this kind of analysis are not even prepared to be run on these large data sets. Okay, so that's why we decided to do the meta-analysis. This is the general single-cell EQTL gen workflow where we have every group has their own data set. We provide, the consortium provides the different pipelines. We're specifically involved in the generating the pipeline for the pre-processing. And in the end, we have the EQTL summary statistics that we meta-analyze, and we get, in the end, single-cell EQTL mapping. But for the project that I want to talk about today, what we wanted to do is to do differential gene expression in this context of the meta-analysis. And the first thing that we wanted to do is to decide what kind of meta-analysis are we going to do. So when we think about meta-analysis, here, for example, I'm showing one data set where we have features. It can be genes, and observations can be either donors or cells, age and sex, and then our summary statistics that we want to measure. And what people normally think about meta-analysis is that we weight our summary statistics by the size of the sample size. So if the data set has a large number of donors or cells, we're going to have a high weighting. And if it has a low number of donors or cells, we're going to have a low weighting. But for our project, we wanted to have a weighting that was per gene. So we decided, in the end, we do this weighting per data set, okay? And then we do the weighted mean, and we get a summary statistic for all. So this is the general framework of the meta-analysis. For our project, we wanted to do a weight per gene, okay? So to do this, what we did, instead of having a simple number, which is the sample size, we just got a vector that is going to allow us to do a meta-analysis based on a gene-by-gene weighting, okay? And basically, the way we do this is we try to see what are the main drivers of these variants of the features that we're trying to measure, and the higher the variance that we see, the higher the weighting. And then we have the slides are weird in the screen. Sorry. Okay. And so what happens is that when we have a weighting per gene, and then we do the weighted mean, and we get a summary statistic that is going to be the weighted by for all the different data sets. All right. So when we do this, we need a proof of principle, right? And we need to do both meta-analysis and reanalysis to see that the meta-analysis actually works. So we use this proof of principle data sets. These are 3 data sets from the consortium that we had access to from Ludwig Franke's lab at Groningen University. The data sets are quite different, so we have different number of donors, different chemistries, and different number of cells. And what we try to do, let me just fix this because, okay. The first thing that we did was to look at sex and age differential gene expression analysis per data set, okay, controlling for batch effects for each of the data set. All right, and these are the results of the meta-analysis. So on the y-axis, we have the number of differential expressions, and on the x-axis, the different cell types. And what we can see is that the meta-analysis have capacity to find more differential expressions. So apparently it has higher power, but I mean, could be real differential expressions or maybe, random stuff. So the first thing that we did then is to do permutation analysis. And I'd recommend to do this permutation analysis on any analysis of this sort that you do in single cell, because in many occasions we've run approaches that we see that you do the permutation and you still get hits, okay? So when you do the permutation, so basically we permuted the level of the donors and we rerun the analysis and the meta-analysis, and what we see, okay, is that we have no false positives, and that the genes that we find that are differentially expressed in these cell types are really, truly differentially expressed genes. And then for sex, it's very easy because we have some expectations of what we should find. We know that males have 1 X chromosome and 1 Y chromosome. So whatever genes that are differentially expressed from the Y chromosome need to be upregulated in males. And this is exactly what we see. These are the genes showed in green here. And in females, we have 2 X chromosomes, but one of the X chromosome is inactivated and should not have expression. But some genes escape this, and these are called escapee genes, and these genes are expressed from both copies of the X chromosome. So if anything, these genes should be upregulated in females, which is exactly what we see here. And then the other interesting thing is that we see a lot of differentially expressed in NK cells. And here I'm showing that these differences are real. And this has been reported before for upregulation in males, but we also see it upregulated in females. And what's interesting about finding differential expressions in different between males and females in the immune system is that females have way more chances of having autoimmune diseases than males. So I think this is what we're trying to figure out, if we can link this to this higher risk of having autoimmune diseases. All right. How does this compare to the reanalysis? Okay, I've said that, if you do meta-analysis, it's faster and you don't have to share data, but okay, are we missing things because we're doing this meta-analysis? So now I'm going to compare this meta-analysis approach with the normal reanalysis that anyone would do when doing differential gene expression analysis, with basically we control for data set. And here are the results. So for sex, we really see no difference, or if anything, with the meta-analysis, we find more differentially expressing. So really, we're not missing much by doing a meta-analysis approach. And in the case of age, the results are very similar. Here I'm showing the results of the meta-analysis, where we see that there is a large signal in CD8 T cells that has been reported before. I'm not going to show this, but the permutations and the analysis, both the permutation and the comparison between meta and reanalysis show very similar results. So the meta-analysis works. We also see high overlap. Here I'm showing the odds ratio with differential expressions that have been reported in another study with lower sample sizes. But one thing that was bugging us is that when we look at the average expression levels of the differential expressions in CD8 T cells, where we have many, it looks like there is some kind of stratification where we find that the genes are especially upregulated in a group of cells and downregulated in another group of cells. And this really looks like changes in cell type composition. So, and indeed, when you look at the subcellular cell types such as CD8 naive, CD8 memory, and so on and so forth, this is exactly what we're seeing. So really, when we work on single cell, if you don't go and look at higher resolution cell types, you might be confounded by cell type composition changes. So the next question is, okay, can we look at cell type composition changes using this meta-analysis approach? So we did exactly this. We modeled cell type proportions and asked, are there differences with sex and age in cell type proportions in these 3 data sets? And basically, in this plot, what I'm showing is the results of this meta-analysis, where in light green, we have the cell types that is not significant, so they don't have significant cell type proportion differences. And here are the results of meta-analysis, where we find more cell types that have cell type composition differences. And in the case of age, we find only one case that is a low number of cells that has change in cell type composition. But what we really care about is not this resolution where you have general CD8-4, CD8 T cells, but rather zoom in and go to the high resolution cell types. And if we do that and we look at CD8 T naive and CD8 T memory, what we see is that CD8 T memory have increased cell type proportions and CD8 T naive have decreased cell type proportions. So really, the results that we were seeing earlier on this analysis were confounded over a combination of changes in cell type proportions and gene expression. And I'm emphasizing this, it can be very obvious, but in most of the analysis that we've seen in PBMCs, and this is true also for equatorial analysis, most of the analysis, because we are limited by sample size, are done at this level. So even though we have single cell data, we can still be confounded by cell type composition. And what we can do to tackle this, right, is to do this differential gene expression at the high resolution, where we can see here that indeed the changes in gene expression that are happening are especially happening in CD8 naive in aging. Okay, and the last analysis that I want to talk about, these are results that we generated 2 weeks ago, so bear with me. But I'm especially interested on this because it's really an analysis that we can only do if we had single cell data. Okay, the other analysis we could do if we had fact sorting and we had only the cell types. But in this case, we really need the single cell RNA sequencing data set that we are using. And basically what we want to see is, are there differences in single cell variability in gene expression between males and females and as we age. So basically, I'm just showing in this toy example where this gene is not variable at all in terms of gene expression, and in another condition, there is a lot of variability in the gene expression. And these are the results of the meta-analysis for sex, so it's much noisier than the differential gene expression. But what's reassuring to us is that we find more stuff in the meta-analysis for the cell types that we have a higher number of cells and lower number of genes for the cell types that we have low number of cells, which is kind of what you would expect. If we now do the permutations, well, the same with age. Okay, so here is a gene, just to convince you that what we see makes sense. This is a differentially variable gene for age, and what we see is that in young, the expression breadth is quite spread, and in old individuals, the expression is tightened, okay? If we look at the permutation analysis, what we see is that in this case, we still have some false positives popping up, but the majority of the genes that we find might be real because they are outside of the distribution of the permutation, but we need to work on this. But what I wanted to show this because if you do the reanalysis, the results are way worse. So basically, the green dot is falling inside the distribution of the permuted labeling results. So basically, the meta-analysis seems to be more robust in this kind of setting. All right, so I have one more slide. And okay, so these are the results. So we're still working on it, but it's interesting to see that we find different sex variable genes and that these genes are biased towards specific cell types. So we find genes that are more variable in females in different cell types and genes that are more variables in males in other cell types. And what was interesting is that we see this in sex. In age, we have a similar number of differentially variable genes, and there's been a lot of literature speculating that maybe as you age, there is going to be more variability in gene expression because we are less able to control for gene expression. But in fact, when we look at age, we see that only in one cell type we find differences, and the differences are towards having more variability in young individuals in CD4 T cells. So we'll explore this more, but I think these results are quite interesting. All right, and I just want to thank my lab, especially Aida. She's here. She is the one that spearheaded the single cell analysis, and she's an amazing PhD student, and also Maria Supena, who also helped a lot with the final results. I also want to thank my collaborators, especially from the Single Cell Equity Gen Consortium, Monique, Lude, Mark-Jan, and Martin, and my sources of funding, and you for your attention. Thank you. Okay.
So with your meta-analysis work, what if I put in, add in another condition, like epigenetic condition, like diet? Would this be?
So you can do it, yeah. You can do any phenotype you want. So we did sex and age because you have this information for all the samples.
But do you think, like in terms of like the gene variability, would that?
Maybe. It would be interesting to see stuff like smoking, for example, where you would have maybe, yeah, that's a very interesting point.
Or exercise for that matter. Right?
Yeah, that would be awesome to have this kind of information. Yes.
Okay. Well, thank you. I wanted to thank all of our